Hello viewers, I am Dr. Lobule. I work as a lecturer of pathology in a medical college hospital and I am making this video for my students and also for you. Hope someone finds this helpful. I am happy to announce that our channel has crossed 50,000 subscribers, so a very special thanks to all our viewers and subscribers for being part of this fascinating journey. Today's topic is T-cell development and thymic education of T lymphocytes. In this video, we will learn about the definition of lymphocyte development, followed by brief discussion on overview of lymphocyte development, and since thymus plays an important role in T-cell development, so we will also talk briefly about the thymus. Then we will also talk about alpha-beta T-cell development, gamma-delta T-cell development, and we will finish our discussion today by talking about NK T-cell or natural killer T-cell development. Okay, a lot of topics, so let's begin. So first we will talk about some introductory points, and always remember that development and differentiation of lymphocytes are tightly regulated by our adaptive immune system. Self-reactive T cells and B cells are removed before they become fully functional. So how can we define lymphocyte development? The process by which lymphocyte progenitors in the thymus and bone marrow differentiate into mature lymphocytes that populate peripheral lymphoid tissues is called lymphocyte development or lymphocyte maturation. Okay, so I am repeating this definition one more time for my students and then we will move on. So how can we define lymphocyte development? It is the process by which lymphocyte progenitors that are located in the thymus and bone marrow differentiate into mature lymphocytes that will populate peripheral lymphoid tissues. So now that we have defined lymphocyte development, now let's move on and talk about the overview of lymphocyte development. Now always remember, it all begins in the multipotent hemopoietic stem cells in our bone marrow. Multipotent hemopoietic stem cells in our bone marrow will give rise to common lymphoid precursors of lymphocytic lineage. Then those cells will either migrate to the thymus to give rise to T-cell lineage of cells, or they will remain in the bone marrow to give rise to B-cell lineage of cells. Now the last bullet point is very important. Always remember, development of T-cell involves sequential rearrangement and expression of T-cell receptor genes, cell proliferation, antigen-induced selection processes, and commitment to subsets that are distinct both phenotypically and functionally. So in this image we can see the overview of lymphocyte development. Now always remember, this is a very simplified diagram. A lot of steps are not shown here to keep this simple. So on the top we can see multipotent hemopoietic stem cell, and from there we can get myeloid stem cells or we can get lymphoid stem cells. And initially the lymphoid stem cells, they are not committed to any lineage. So that's why in the previous slide we had mentioned common lymphoid precursor. But ultimately they will become committed, so some of them will make B cell, and from B cell we will ultimately get plasma cell, and again some other lymphoid stem cells will be committed to make T cells, and from there we will also get T helper cell and also cytotoxic T cells, and we can also see that some of the lymphoid stem cells will be ultimately committed to produce natural killer cells. So this is a simplified overview, and we will talk in details in the later half of today's video regarding the steps that are involved in T cell development. So don't worry. Now, T lineage cells will differentiate along one of three developmental pathways within the thymus. Some of them will differentiate into cells that will express alpha-beta T cell receptors, other will express gamma-delta T cell receptors, and others will differentiate into cells that will share functional properties of natural killer cells, and those cells will be called natural killer T cells, as we will see. Now always remember, T cell development is happening inside the thymus. So now let's talk briefly about the thymus. So thymus is a lobulated primary lymphoid organ. Now, whenever you mention this term, primary lymphoid organ, the examiners are very fond of asking you, what are the primary lymphoid organs? Always remember, bone marrow and thymus, these are our primary lymphoid organs. So, thymus is a lobulated primary lymphoid organ. It has very important role in cell-mediated immunity. Embryologically, thymic epithelium is derived from third and inconstantly from fourth pair of pharyngeal pouches, and these are endodermal derivatives. However, thymic lymphocytes are mesodermal in origin. So the weight of the thymus at the time of birth is from 10 to 35 grams. Then it grows until puberty to gain maximum weight of 20 to 50 grams. After puberty, it progressively involutes, and so in older adults, it weighs little more than 5 to 15 grams. Now, thymus is bilobed and encapsulated, and fibrous extensions called trabeculae will extend inwards from the capsule, and they will divide each lobe into multiple incomplete lobules. Now this term is very important, incomplete lobules, because since the lobules are incomplete, the medulla will show continuity between the neighboring lobules. And always remember, each lobule will have outer cortical layer and central medulla. So here I have drawn a very simple diagram of the thymus. So we can see the capsule, we can see trabeculae that are extending from the capsule inwards, and we can also see the cortex that is drawn in blue color, and we can also see the medulla that is drawn in pink color. And since the lobules are incompletely divided, so we can see that there is continuation between the medulla of various lobules. Now, thymus is populated by diverse types of cells. Prothymocytes are T cell precursors that will migrate from the bone marrow to the thymus, attracted by thymic molecules like lymphotactin. Upon entering the thymus at the corticumedullary junction, they are named thymocytes. The thymocytes will then undergo a strict positive and negative selection process as they migrate from the thymic cortex to the medulla, driven by chemokines. Now, this second bullet point is very important. Pay attention here. Only 1 to 5% of all thymocytes will pass the selection test to graduate as T lymphocytes. So what will happen to the remaining 95 to 99%? The other 95 to 99% will either die by apoptosis after failing one of the selection tests, or they will leave the thymus before undergoing selection. And the last stem, that is leaving the thymus before undergoing selection, is particularly true for gamma delta T cells, as we will see. Now, other cells in the thymus are called epithelial reticular cells, and they will include dendritic cells, macrophages, and epithelial cells. Dendritic cells derived from bone marrow, and they are present at corticumedullary junction and also within the medulla of the thymus. Macrophages are primarily present in the medulla. The epithelial reticular cells will serve as instructors for the thymocytes. Now, in the thymus, you will also find this structure that is called Hassel's corpuscles. And always remember, these are oval structures consisting of round or spherical aggregation of flattened epithelial cells, and they are composed of central mass and a capsule that is formed by epithelioid cells. So now that we have talked about the thymus, now let's move on and talk about alpha beta T cell development. Newly derived cortical thymocytes are CD4 negative, CD8 negative, T cell receptor negative, as well as CD3 negative. Since they do not express CD4 nor CD8 on their surface, so they are also called double negative or DN cells at this stage. What will happen next? Soon they will generate and express CD4, CD8, CD3, and also they will recombine their T cell receptor genes to generate unique T cell receptors. Since now they are expressing both CD4 and CD8 on their surface, so now the developing cells will be called double positive or DP cells. Now, double positive thymocytes will undergo a strict positive selection test, and those failing the test will die by apoptosis. So what do we mean by positive selection test? It is the process in which thymocytes whose T cell receptors bind with low avidity to peptide-self MHC complex are stimulated to survive and to differentiate either into CD4 positive or CD8 positive T cells. Now, I have two separate videos entirely on MHC molecules or major histocompatibility complex molecules, so you can also watch those videos after finishing today's lecture to know more about MHC molecules. Now, double positive cells that are failing this step can make further attempts because all cells have maternal and paternal alleles of their T cell receptor genes. Double positive cells stuck at this stage can continue to mutate and re-audition T cell receptor genes to make more suitable T cell receptors. However, as we have seen, most of the double positive cells will fail this step and ultimately they will die by apoptosis. So that is written in the third bullet point. Double positive cells that are failing to recognize and bind to self MHC molecules or that are failing to bind with self MHC plus peptide molecules that are expressed by cortical antigen presenting cells will die by apoptosis within 3 to 4 days. Always remember, this type of death is also called death by neglect. Now, during transition from double positive cells to single positive cells, thymocytes whose T cell receptors can recognize self class 1 MHC molecules become CD8 positive and CD4 negative cells. Similarly, thymocytes whose T cell receptors recognize self class 2 MHC molecules become CD4 positive and CD8 negative cells, and this is sometimes referred to as rule of 8, because we know that 8 multiplied by 1 is 8, and also 4 multiplied by 2 is 8. And you can use this to remember that CD8 positive cells can recognize class 1 MHC molecules, and CD4 positive cells can recognize class 2 MHC molecules, as we will see. Cells passing the positive selection test and located at the corticumedullary junction are allowed to enter the medulla. So, what is the importance of positive selection test? It guarantees that T cells that eventually exit the thymus have functional T cell receptors that can bind to self MHC molecules. Survivors of positive selection will undergo a second selection test, that is called negative selection, when they arrive at the corticomedullary junction. Here they will interact with second sets of epithelial reticular cells that are expressing self-antigens. Now, you may be asking me, Dr. Robiul, how does the thymic epithelial cells express self-antigens that normally should not be in the thymus? And that's a very good question, and the answer lies in a fascinating nuclear protein called autoimmune regulator. So let's talk about that now. Now, in the medulla, thymic epithelial cells express a nuclear protein that is called autoimmune regulator, or AIRE. Now, this nuclear protein can induce low-level expression of many antigens that are normally expressed only in specific peripheral organs. Tissue-restricted self-antigens can be expressed in the thymus due to their action. Thymocytes, whose receptors can recognize peptide MHC complexes with high avidity, will undergo apoptosis. This is negative selection, or they may differentiate into regulatory T cells. So the last bullet point is very important, because here we can see that thymocytes, whose receptors are recognizing peptide MHC complex with high avidity, they are either dying by undergoing apoptosis, or they are getting differentiated into regulatory T cells. So what is the importance of negative selection? It ensures that naive T cells that will exit the thymus are not specific for self-antigen, because if they were specific for self-antigen, then that would have resulted in autoimmune diseases. Thymocytes that pass both positive and negative selection tests have graduated from the thymus. They will now enter the circulation through the medullary postcapillary venules as T cells. So here we can see a simplified image that is showing how we are getting various types of T lymphocytes from the T cell precursors. So on the far left we have the T cell precursor that was present in the bone marrow. Then those pro-thymocytes went to the thymus, and there, upon entering the thymus, they became thymocytes, and initially they were double negative and then they became double positive. When they became double positive, they are expressing both CD8 and CD4 on their surface, and ultimately they are differentiating into single positive T cells. Some are becoming CD8 and some are becoming CD4 positive T cells, and from the CD8 positive T cells we will ultimately get cytotoxic T cells, and from CD4 positive T cells we will get helper T cells, and again helper T cells will be further differentiated into Th1, Th2, Th17, and also into regulatory T lymphocytes, as we will see. And in these differentiation processes, there will be important roles of different types of cytokines. So you can also see that I have also summarized those cytokines in this image as well. So you can pause this image to memorize those cytokines because they are important for your multiple choice questions. So now that we have talked about alpha beta T cell development, now let's move on and talk about gamma delta T cell development. In the fetal thymus, first T cell receptor gene rearrangement involves the gamma and delta loci. In developing double negative T cells, initially rearrangement of T cell receptor beta, gamma and delta is possible. If a cell succeeds in productively rearranging its T cell receptor gamma as well as its T cell receptor delta loci before making a productive T cell receptor beta rearrangement, then it is selected into the gamma delta T cell lineage, and this occurs in about 10% of developing double negative T cells. Many of these cells will fail to express CD4 and or CD8, so they will not undergo positive and negative selection tests like the typical alpha beta T cells. They will depart from the thymus shortly after developing their T cell receptor complexes. So what are the roles of these gamma delta T cells? These T cells are thought to be transitional cell types that may be a bridge between innate and adaptive immune system. The last topic that we will talk about briefly today is regarding the NK cells or natural killer T cells. Now always remember, natural killer T cells share some characteristics with natural killer cells. They express several surface markers and receptors that are found on natural killer cells. However, they also undergo some development in the thymus and they also express T cell receptors. So that's why we are calling them natural killer T cells. And the last bullet point is particularly important for your multiple choice exams. They can specifically recognize epitopes that are presented by non-classical MHC class 1 molecule called CD1D. So this concludes our lecture on T cell development and thymic education of T cells. I hope this video was helpful. If you like my videos, do comment, share, subscribe and let me know. And for my students, I will also recommend you to go through your textbooks to know more. Okay, that's all for today. Until next time, take care and stay blessed. Thank you.